several big things. So, first, finally got all the problem-solving videos posted last night. Been having camera problems. That's been part of the delay, but they are now all posted and hopefully helpful to you. If you have questions about those, please let me know. Second, not a surprise to you, we have an exam on Friday. I hope it's not a surprise to you. The exam is covering materials since the first exam. I have scheduled a review session for tomorrow night at 5 o'clock. Tomorrow at 5 in ALS 4001. I will videotape it, so if you can't make it, I'll be there with usual with my camera. That hopefully is working. Now, the camera, people saw that the outline or sort of overview video that I had stopped in the middle of me talking about the epidermal growth factor. That was because the battery ran out, and when the battery ran out, the whole camera has gone dead on me, which is why I'm having some camera problems. I've got a backup camera today I think will hopefully take care of things, and that should be all set. I will review that epidermal growth factor stuff a little bit at the beginning of the review session on Tuesday, so everybody has similar access to that. But like the other review sessions, other than that, it will be driven by your questions. Okay? Third or fourth, I've lost track. But whatever number I'm on, I have decided that the exam will not cover glycolysis. So, where I finish with metabolic energy, that's where the exam will cover. Everybody's fist bumping, okay. Like they did something, right? Oh, we did it, guys. We're number one. Oh, wow. I don't know. In any event, you're number one, all right? So glycolysis will not be on the exam, okay? All right. And I think that's the end of my announcements. Are there any questions with that? Hearing none? Everybody's ready to finish up metabolic energy. Okay, so metabolic energy, as I said last time, is obviously important for us to consider because some processes require energy, and we have to make sure we generate sufficient energy for those processes. And other processes generate energy. They release and make ATP, and they make it possible for those requiring processes to run. One of the guiding principles to remember about metabolic energy is that metabolic energy, okay, when we look at, if we run a cycle like, let's say, glycolysis where we break down sugar, and we look at how much energy is released and captured in the form of ATP, and we compare it to the process of making that same sugar, it will always take more energy to make than we will get out of it in breaking it down. That's because no process is 100% efficient, and some of that energy is lost as heat. One of the reasons we generate heat when we exercise is because that's a reflection of that loss that's happening during that generation process. Okay? For that reason, cells don't want to have catabolic processes going on at the same time as anabolic processes unless they want to generate heat. Now, in some cases, they actually use that as a means of generating heat. All right? But 99% of the time, they do not want to do that because it's nothing but a waste of energy for cells, cells to go through a process like that. All right. So keep that in mind as we get talking about metabolism, which we're going to be diving into starting later today. Okay, now another guiding principle for metabolic processes is what I call coupled reactions. Now, coupled reactions occur when an energy yielding process is combined in a reaction with an energy requiring process. If you look on the screen, you see what it takes to put a phosphate onto glucose. If I put glucose and phosphate together and I measure the energy necessary for that process to happen, you can see that the delta G0 prime is plus 14 kilojoules per mole, meaning that if I start with equal concentrations of glucose phosphate and glucose 6-phosphate, that at equilibrium the reaction will shift to the left. if I start with equal amounts. Okay? Now, that means that that is energetically unfavorable for that reaction, and it's what I like to think of as an energy barrier. Notice we're talking about the delta G0 prime, not the delta G. Okay? If I take, and in fact cells don't put phosphate onto glucose in this way because it's not practical for them to do that. They would get very, very little phosphate onto glucose if this was the way that they phosphorylated glucose. This reaction is actually the first reaction of glycolysis. Okay? Instead, cells use the second reaction. The second reaction, they couple the hydrolysis of ATP with the transfer of phosphate to glucose. So instead of using inorganic phosphate, all right, we're using the energy of ATP. And you remember from last time when I talked about ATP, those phosphates repel each other. That's actually the source of all that potential energy that's in ATP. Cleaving that bond releases that energy. And the release of that energy is coupled to the movement of that phosphate onto glucose to make glucose 6-phosphate. If we couple the hydrolysis of ATP to the phosphorylation of glucose, we see that that is an energetically favorable process. The delta G0 prime for that is minus 16.5 kilojoules per mole, meaning that if we start with equal concentrations of glucose, ATP, and glucose 6-phosphate, that at equilibrium, that reaction will be far to the right. Okay? At equilibrium, if we started with equal concentrations, at equilibrium, we will have the reaction go far to the right. So, coupling of energetically favorable processes with energetically unfavorable processes makes reactions go. And that principle occurs over and over and over in metabolism. It's actually in this way that ATP is used to make molecules. It's this way that ATP is used to make molecules. Sometimes people think, okay, well, you just cleave ATP and this energy is magically released and energy magically captured to do something. That's not how it happens. It's not like lighting a fire with gasoline, for example. Okay. Now, just so I can scare you a little bit, there's metabolism. This is what we're getting into, guys. Is that a scary thought? What's that? Is that a scary thought? It's not scary, all right? I show it to you actually not to scare you, but rather to show you the way that metabolism really resembles a roadmap. Metabolism looks like a roadmap, and each one of those little dots on there is a town, and those little pathways that connect are individual molecules, that is, individual steps in that pathway. And when we think about it this way, we can really get from anywhere on this pathway to anywhere else by following multiple different paths. Just like we have multiple ways we can get to Portland, we have multiple ways we can get from glucose to acetyl-CoA, for example. Okay? There's many ways in which that process can happen. And it's important to remember that. Okay? We see also with these pathways that these pathways are interconnected. All right? The very central part, if you look at it carefully, specifically, right in, I don't have it here, but specifically in the central part of it, where the up and down part is, you see glycolysis and gluconeogenesis. Those two things are what we call central metabolic pathways. They're central because they're central to virtually every cell on the face of the earth. Virtually no cells, in fact, I know of no cell that skips all of the reactions. There's a few cells around that have some unusual ways of bypassing those reactions, but no cells completely bypass all the reactions of glycolysis and gluconeogenesis. Okay? So they're central to everything that happens. The citric acid cycle, which is shown in the lower left, although it's not shown centrally, is also something we call a central metabolic pathway. Now, those links that we see between them, and they've tried to use color in these cases to indicate molecules of a common theme, for example. If we look at those molecules connecting pathways, we realize that the products of one pathway are the starting materials for another pathway. Another thing I want you to take away about metabolism before we actually start talking about it, and this one I'm going to remind you of over and over, and that's the fact that metabolism does not occur in isolation. It does not occur in isolation. We think of metabolic pathways as, well, here's glycolysis, and it's separate from everything else. And glycolysis is not separate from everything else. The intermediates of glycolysis are found in a variety of places. We'll see glucose 6-phosphate in just a little bit is connected to many pathways. So when I see a glucose 6-phosphate, I can't look at it and say that's in glycolysis because it could have been produced by any number of pathways. Does that make sense? The only sequestration of materials that we see in cells is in eukaryotic cells where some reactions are sequestered in organelles. Fatty acid, fatty acid oxidation, for example, occurs in the mitochondrion. The citric acid cycle occurs in the mitochondrion. Glycolysis occurs in the cytoplasm. So there is some sequestration that happens, all right? But I can't say that glycolysis is separate from everything else because the pentose phosphate pathway, which you can see on the screen, occurs in the cytoplasm as well. So they're not isolated pathways. Please remember that. Please remember that metabolic pathways are inventions of human beings. They're ways for us to group things, all right, and say, well, this group of 10 reactions is glycolysis, but in the cell, there's no such thing as glycolysis. They're not separate from everything else. They're all part of that bigger picture. Okay. This scheme shows a little bit more clearly, I think, how the pathways are interconnected. We can see, for example, that sugar metabolism is feeding the citric acid cycle. And that's what happens. Glycolysis makes products, or makes a product that is used to produce materials to enter the citric acid cycle. And then those materials in the citric acid cycle get oxidized. So where you see these arrows connecting individual pathways that are here, these arrows are indicating molecules produced by one that are going to another pathway to be reacted upon. We see, for example, fats and fatty acids are linked. No surprise. When you break down fats, you get fatty acids. You see fatty acids go to prostaglandins, or the prostanoids, as I've talked about. Okay? That's how prostanoids are made. We see that amino acids are very tightly linked to the citric acid cycle. When you break down protein, when you're on that low-carb diet, the energy that you're getting is coming from amino acids, and those amino acids are partly being oxidized in the citric acid cycle. That's how you're getting biological energy when you're on a low-carb diet. It's one of the ways. Okay? So we see the interconnectedness of these pathways and realize that metabolism is really a very complex picture. Now, we're not going to deal with the complexity of the kinetics of that picture. That would be beyond this class, and it would be more of a topic for a graduate level class than it would be for an undergraduate class, especially for non-majors. But I want you to keep this image in your head in terms of how pathways are interconnected. And no, I'm not going to ask you to draw this picture. I'm not going to ask you to draw this picture. Okay. I promised you I would show you glucose 6-phosphate occurring in several pathways. We see at the very top the breakdown of glycogen can produce, ultimately, glucose 6-phosphate. We see the glucose 6-phosphate can be used in glycolysis. We see glucose 6-phosphate can be used in the pentose phosphate pathway. We see the glucose 6-phosphate can be used to make glucose. Okay? This is just one example, but there are many examples of molecules within a pathway that are used for a variety of purposes. Aspartic acid is a really good example. Aspartic acid is an amino acid. If I transaminate aspartic acid, okay, I end up with oxaloacetate. And no, I'm not going to ask you to tell me that. I'm just telling you for example. Oxaloacetate is used in the citric acid cycle and can be used to derive metabolic energy. That's how you're getting one of the ways you're getting energy from amino acids. Okay? So remember this connectedness. Very, very important thing. Look at the pathways mentioned here. Gluconeogenesis, citric acid cycle, glycolysis, pentose phosphate pathway, and glycogen metabolism. All of those pathways are related to the metabolism of glucose 6-phosphate. Okay, well, I'm going to show you this figure a second time in just a second, but I conclude this discussion of the metabolic pathways by drawing you an over... Yes, question? Why were the red and green labels on the... Oh, wrong one. On the last one? I was using, in this case, the green to indicate pathway names. I drew this figure, so they're just simply indicating pathway names, and red was indicating enzymes. So you can see all the enzymes had an ASE, or they're all named as an ASE at the end. Yeah, nothing specific, or nothing profound, but that's what I was doing. Okay, here's an overview. For the next couple of days, we're going to be dealing with glycolysis, the very top part of that process where we go from glucose to 2-pyruvates. All right? We're going to spend a couple of days going through that process. And we see that pyruvate leads to other things. We see pyruvate's got 3 arrows coming off of it. Okay? Pyruvate has 3 different fates relating to the oxidative metabolism. Okay? It has other fates if we talk about amino acid metabolism. So like glucose 6-phosphate, pyruvate is a very central molecule in a variety of processes, and we'll talk more about that. We also see that glucose is ultimately feeding the citric acid cycle, as I mentioned to you before. And the citric acid cycle, as we will talk about next term, is a pathway that is very important for oxidative metabolism. Very important for oxidative metabolism. When we talk about metabolic energy, the vast majority of our metabolic energy comes ultimately from the citric acid cycle. So we're going to spend a lot of time talking about that. That will occur at the beginning of next term. Okay. I forgot I had this. All right. So one of the things that oxidation produces are high-energy intermediates. And the high-energy intermediates are quite high energy in some cases. If we look at the energy in ATP, for example, comparing the breakdown energy of ATP to ADP, we see we've got minus 30.5 kilojoules per mole. That's a pretty fair amount of energy. In fact, when we talk about that reaction, we realize how much potential energy can be put into a reaction to make it happen. We saw that with making the glucose 6-phosphate, adding ATP really made a difference in terms of how much energy was coming from ATP. And that's really how much you get. There are other molecules, interestingly enough, that have more energy than ATP. Last time I talked about creatine phosphate as one of those molecules. And you can see creatine phosphate has minus 43.1 kilojoules per mole. A lot of energy there. But there's some that have even higher. Right? One of those, 1,3-bisphosphoglycerate, an intermediate in glycolysis. Another one, PEP, phosphoenol pyruvate, another intermediate in glycolysis. We see that PEP has essentially as much energy as two ATPs. And it's because of this that we see in glycolysis that we create what I call the Big Bang. It's a Big Bang when PEP gives up its energy. And we'll discuss that more. Okay? Now, we have high energy molecules, but the way that we make ATP is primarily not from the release of energy by high energy molecules. That won't become apparent until next term. We will see some examples where high energy molecules are used to make ATP in glycolysis. This is a process called substrate level phosphorylation. But it's a relatively minor way for our cells to make ATP. Okay. And let's see, am I caught up there? Yes. Okay, so that's where we finished last time. Any questions on that before I jump into glycolysis? Question? I'm sorry, I can't hear you. What's the difference between substrate level phosphorylation and oxidative phosphorylation? So substrate level phosphorylation will always include a high energy molecule that's used to make ATP. And oxidative phosphorylation involves electron carriers in the mitochondria, and we'll talk about that next term. Okay? Yep. They're very different processes. For many years, people thought, all the way up until the 60s, people thought that all ATP was made from high-energy molecules, and they couldn't find sufficient quantities of high-energy molecules to explain how much ATP was being made in a cell. It was quite a puzzle for a long time until they finally figured out that they were completely missing oxidative phosphorylation. And I'll talk about that, as I said, next term. Other questions? Okay. So, where am I? All righty. The biochemistry of carbohydrates. So, what we're getting ready to talk about, and this is where the material for the exam 2 will end. Okay? So, you're going to start the material for the final exam at this point. All right? Okay. What we're getting ready to talk about, let's move this guy over so it shows up on the video. And, by the way, I know that many of you are liking the outlines, and I have to say I am too. It's helping me to get things a little bit more organized for you. So that's been a good exercise for both of us. Yes, question? I thought I did. Are they not online? If I've overlooked some, I'll make sure I get them posted. I thought at least I'd gotten some of them up there. If not, let me know. But yeah, you're welcome to them. Okay. The biochemistry of carbohydrates. So, here's that figure I showed you last time. And again, I show it to you just so that you keep in mind that overall process. If we look at the overall oxidation of glucose, we start at the top at glucose and we get down to the very bottom and we see we produce 6 molecules of carbon dioxide and 6 molecules of water. That's what we get from the complete oxidation of glucose. In addition to that, if we run that completely through the oxidative phosphorylation process and the citric acid cycle, depending upon how people count their ATPs, you get between 30 and 38 ATPs. Okay? If you go through the processes shown above it, that is anaerobic fermentation or for yeast and bacteria, or anaerobic fermentation for animals, the grand sum of product you get is 2 ATPs. 2 ATPs. So you're going to produce a hell of a lot more ATP energy when oxygen is present than when oxygen is absent. Therefore, when you go get aerobic, that's the most efficient way to lose weight. Is that right? How many say yes? How many say no? Okay. Anybody want to say why they say no? Well, I think it's no. He must be. People are trying to outguess me, right? Yeah. It's correct. Okay? If you want to lose more weight, you want to go anaerobic, not aerobic. All right? Because you get less ATP per glucose broken down, means you're going to break down more glucose, right? When you think about getting aerobic, everybody is thinking, oh my goodness, this is the way to go, because the term is misused, folks. When you say you're getting aerobic, you're actually getting anaerobic. Your body's breathing heavily because it's trying to catch up from being anaerobic. That's why exercise makes a difference. Okay? So the next time you see somebody talking about being aerobic, you can remind them that they're really becoming anaerobic, and that's probably better for them. Okay? Make them think about that one for a while. Okay. So, If you're going to make the most ATP, you're going to want to be aerobic, and the cells most of the time are aerobic. All right? Only when we're exerting ourselves very heavily do we get much into the anaerobic phase. We'll talk about that later. Here's the process of glycolysis. Glycolysis has 10 reactions. You can see the 10 reactions on the screen. And we start with the 6-carbon molecule glucose at the very top. By the middle, where we get to the DHAP and the GA3P, these are 3-carbon molecules. And fortunately for students having to memorize pathways, the two 3-carbon molecules become the same one. They become 2 molecules of glyceraldehyde 3-phosphate, or 2 GA3Ps. I'm not going to make you memorize the structures of glycolysis. You should know the structures of things that make sense. For example, you already have to know the structure of glucose. It's not unreasonable for me to expect that you would know the structure of glucose 6-phosphate or fructose 1,6-bisphosphate, because you already know the structures of glucose and fructose. Okay? You should know the names of the enzymes of glycolysis. Okay? Now, the names of the enzymes of glycolysis are very simple because the name, in all but one case, tell you exactly what the reaction is doing. The first enzyme of glycolysis is called hexokinase. Look at the name. Hexo meaning 6, kinase meaning phosphate. You're putting a phosphate onto a 6-carbon molecule. That's what hexokinase is doing. The second enzyme is phosphoglucoisomerase. Phosphogluco, meaning you're starting with a phosphorylated form of glucose. Isomerase, you're rearranging it. The second reaction is rearranging glucose 6-phosphate into fructose 6-phosphate. So if you know the enzymes, it actually tells you what's happening in the pathway. So I will expect that you will know that. Now I'm going to take you through the individual reactions and tell you something about each of the reactions, their significance, and some considerations for them. When we look at metabolism, it's important to remember a couple of things. Yeah, the reactions are important, all right? But more important is how a pathway is controlled. How a pathway is controlled. Because this is an energy-releasing pathway. It's an energy-releasing pathway. It's important for the cell to capture the energy of this pathway, make ATP, and this pathway also makes NADH. Okay? We want to control this pathway because we don't want to be breaking down glucose, for example, when we have very little glucose. We don't want to be breaking down glucose as our cell is struggling to make glucose. So that control of the pathway is every bit as important as the reactions of the pathway. Now, glycolysis turns out to be a really unusual metabolic pathway. I talked about pyrimidine synthesis and the control through ATCase. And when I talked about ATCase, I said we had feedback inhibition. The end product, CTP, inhibited the first enzyme, ATCase, and the whole pathway was stopped. Glycolysis turns out to have a more complex regulation. There are actually 3 enzymes in glycolysis that are controlled. 3, not 1. And one of those enzymes controls the very last step in the process. And that's an interesting phenomenon, as we shall see. Okay? So glycolysis is not your typical metabolic pathway. It's very unusual in that respect. Okay. All right. So glycolysis is the primary energy source of cells, ultimately. We derive more energy from fat than we do from glucose. But fat is a storage form of energy. Glucose is an immediate form of energy. Glucose is dissolved in the aqueous environment of our cells. It travels through our blood very easily. And it's because of that when we have an emergency, the body releases glucose quickly, not fat and fatty acids, because they don't travel in the bloodstream very easily. So glucose is the primary energy source of cells, and that's ultimately coming through this process of glycolysis. It is what I call a central metabolic pathway. We see that all the other pathways depend upon glucose or glycolysis ultimately. All the reactions of glycolysis, all 10 reactions, occur in the cytoplasm of cells. Okay? And finally, there's 2 phases to the process. The first phase is what I call an energy investment phase. The energy investment phase actually requires ATP to start the process. So here's a pathway that's going to ultimately produce ATP, but it needs ATP to start the process. The second part of the process is the energy production phase. Okay? Looking at this pathway, if we do add up all the numbers, we see it takes 2 ATPs to start the first part of the pathway. And in the second part of the pathway, we get a total of 4 ATPs produced. So the pathway is producing a net of 2 ATPs. It's also producing 2 molecules of NADH because we have 1 oxidation occurring in glycolysis. 1 oxidation in glycolysis. Most of the oxidation of glucose does not happen in glycolysis. Most of the oxidation of glucose happens in the citric acid cycle. Okay. So let's go through the reactions. All right? Well, actually, before I do that, just a little background. So glucose is stored as polymers in cells. If you're a plant, it's stored, as I've noted before, as amylose or amylopectin. Those combine to make starch. In animals, we store glucose as glycogen. Is that a question? Oh, you're just waving at me. Okay. Hello. Okay. All right. So glucose is a poison. So we don't have a lot of free glucose lying around. Cells sequester it as glycogen or as amylose, amylopectin. The good thing about glucose is it's very easily released from polymers. Very easily released. That's particularly important for animals that need very quick sources of energy to run, to jump, to hide, to scream, to do whatever it is that they need to do. Glucose travels easily in the blood, as I've said. And glucose is made from very simple precursors. We'll see that in the process of gluconeogenesis that I'll talk about most likely next week. Okay, let's get into the reactions. Here's the first reaction of glycolysis. It's catalyzed by the enzyme known as hexokinase. You've already seen it, and I've talked about it numerous times during the term, actually. In this reaction, a phosphate is being put onto carbon number 6 of glucose. We can see quite easily what's happening with that. Okay? There's carbon number 6, there's the phosphate, and bang, the phosphate goes on to carbon number 6. Okay? We saw earlier that was an energetically unfavorable process. If we simply used phosphate, it's energetically favorable process when ATP is used as the source of the phosphate. This is the first regulated step of glycolysis. Hexokinase is a regulated enzyme. Its regulation is unusual, and I don't say too much about it. I'm going to tell you a brief thing about it, but I really won't focus on hexokinase because it's not the most important regulated enzyme in glycolysis. Hexokinase is actually inhibited by its product, glucose 6-phosphate. When glucose 6-phosphate accumulates, it inhibits the enzyme. Now, this sort of makes sense. If glycolysis is getting stuck, what's going to start happening? We're going to see accumulation of intermediates. When is glycolysis going to get stuck? Well, how about when the cell has plenty of energy? If the cell has plenty of energy, it doesn't want to be breaking down glucose. You don't want to be lighting a fire in your fireplace in July if firewood is going to be limiting in the winter, right? So you don't want to be burning glucose if the cell has plenty of energy. So having something that inhibits hexokinase to start the process when glycolysis starts backing up makes a lot of sense. Okay. There's hexokinase. I love that picture. Hexokinase is the big guy, and this is drawn to scale. We can see the size of the hexokinase enzyme compared to the substrates that it works on, ATP and glucose on the site. How in the world do these guys find each other and do their thing? Pretty amazing process. Okay? Hexokinase is found in virtually all cells. That's no surprise because glycolysis is occurring in virtually all cells. Hexokinase is not specific to glucose. That's why it's called hexokinase. It will actually work on a variety of 6-carbon sugars. It will phosphorylate galactose, for example. It will phosphorylate mannose. Okay? All it really cares about is that it has a 6-carbon sugar on which to do its thing. There's a related enzyme called glucokinase. It is specific for glucose. Okay? And it's found in the liver. Now, when I talk later about the Cori cycle, I'm going to talk about this enzyme. I want you to plant this enzyme in your head to remember. Glucokinase is an enzyme that is specific for glucose, and it's found primarily in the liver. There's a reason why it's found primarily in the liver, and we'll see that when we talk about gluconeogenesis and the Cori cycle. And I said hexokinase is inhibited by its product. And now we move to the second reaction. The second reaction of glycolysis. Phosphoglucoisomerase. The name is telling us what it does. Phosphogluco, again. Glucose 6-phosphate is a phosphogluco. Isomerase meaning it's rearranging itself. Yes, sir? Is glucose 6-phosphate a heterotropic effector? That's actually a good question. It's not working. I would say it is not a I wouldn't categorize it as a homotropic or as a heterotropic. The way it's affecting the enzyme is not as part of being a substrate. So I would say no, it's not really in either category. It's a very good question. Good thought. Yeah. And hexokinase's regulation is unusual. Okay? And if you want, I'd be happy to talk to you about it later, but it is an unusual enzyme that doesn't fit into that categories that we've talked about before. Okay. This reaction is pretty straightforward. We're converting a glucose 6-phosphate into a fructose 6-phosphate. Okay? Glucose is going into fructose. We see that isomerization that's happening. And by the way, you see a lot of abbreviations that I use. There's G6P isomerase, which is another name for that. You're welcome to use any abbreviation that I use, as I've talked about before. So any abbreviation I use, you can use. You must use my abbreviations. You can't use your own. Otherwise, I say, well, XYZase is not exactly the right name of the enzyme, guys. But that's my abbreviation. Okay. All right. This reaction is readily reversible. The delta G0 prime is a positive 1.7 kilojoules per mole. That's pretty close to 0. Meaning that if we start with equal concentrations of products and reactants, that at equilibrium we'll have pretty close to equal concentrations of products and reactants. It doesn't change much. The next reaction is probably the most important reaction of glycolysis as regards regulation. This reaction is catalyzed by the enzyme known as phosphofructokinase. You're welcome to call it PFK1. PFK1. Okay? We'll see later there's a PFK2 that we'll talk about. But PFK1, okay, is basically putting a phosphate onto position number 1 of fructose 6-phosphate to create fructose 1,6-bisphosphate, or F16BP. Okay? This reaction is very much like the hexokinase reaction. If we try to do it in the absence of ATP, instead just try to put a phosphate on there, we see it's very energetically unfavorable. However, when we use the energy of ATP, the reaction becomes energetically very favorable. This reaction goes forward quite readily. Now, phosphofructokinase, as I said, is a regulated enzyme. We're going to see its regulation is really interesting. And it's very well tuned to cellular energy situations. This enzyme you want to have turned on when cell energy is low, and you want to have turned off when cell energy is high. Okay? We'll see how that plays out as I talk later about the regulation of this enzyme. Here's the reaction. There's carbon number 1 with its hydroxyl. There's the phosphate from ATP. And there's the phosphate put onto it to make the fructose 1,6-bisphosphate. Okay? Very important regulatory enzyme. Look at that delta G0 prime, minus 14.2 kilojoules per mole. we start with equal concentrations, products and reactants, it's going to go far to the right, at equilibrium. Okay. Now, in terms of the regulation, I'm going to talk about it briefly here, but I want you to just kind of plant these ideas in your head. When I talk about the regulation relevant to gluconeogenesis later, I'll talk about these things in more detail and show this to you in a different way. But from a simple perspective, all right, there's some allosteric regulators of the enzyme. AMP and fructose 2,6-bisphosphate, F26BP, activate the enzyme. We'll talk about F26BP later. But you know that AMP is an indicator of low energy inside of cells. Low energy. So low energy makes sense to activate this enzyme. When the cell has low energy, it wants to be breaking down glucose so it can get more ATP energy. And this makes very, very good sense. This enzyme is also, and this is a really interesting thing about this enzyme, inhibited by ATP. Why is that an interesting thing about this enzyme? I know that it's because ATP indicates high energy, but there's another reason why this is interesting and important. Anybody? Yeah? 1 point of extra credit right there. Excellent. Excellent. Okay. ATP is a substrate. Now think about this. Here's a substrate that is inhibiting an enzyme. We've never seen that. We haven't seen this anywhere. My next question to you is, how is it that a substrate is inhibiting an enzyme? Any thoughts? Yes? Could it be like a suicide inhibitor that once it binds, it doesn't let go? It could be, but it's not. And that would probably not be a very efficient way to go because you'd have to make a lot of enzyme to make up for it. Yes? Is it more like an alpha G protein that takes a while to break down? No. One point of extra credit right here. You got 2 binding sites, one for the substrate and one for the allosteric effector. Very good. Okay? Very, very good. See me after class about the extra credit. Yes. That's exactly what's going on. So you got 2 binding sites. One binding site has a very high affinity for ATP, and one binding site has a very low affinity for ATP. Which one would have the high affinity for ATP? The substrate. Because you want to be having this process go when there's low levels of ATP, right? So at low levels of ATP, you want to be able to grab that substrate as best you can. You want to turn this off when ATP levels get high, and that's not going to take much binding affinity for that to happen. So the allosteric binding site is going to have a lower affinity than the substrate binding site is. Okay. 2 binding sites. There we go. Now, the next reaction in the process is a very odd reaction. Very odd reaction. I'm going to say a little bit about it, and I'll probably say more about it to start the lecture next time. It's catalyzed by an enzyme. It's the only one in the pathway whose name doesn't tell you a lot about what it does. The name of the enzyme is called aldolase. Okay? Aldolase catalyzes the breakdown of a 6-carbon molecule, fructose 1,6-bisphosphate, into 2 3-carbon molecules. One is glyceraldehyde 3-phosphate that I've abbreviated GA3P, and the other is dihydroxyacetone phosphate, or DHAP. Okay? Now, what's odd about this reaction is that this reaction is energetically very unfavorable. Look at the delta G0 prime there, 22.3 kilojoules per mole. If we start with equal concentrations of products and reactants at equilibrium, we're going to have the reaction going far to the left. How in the world does the cell overcome such an energy barrier? Well, the cell knows math. That's how. The cell knows math. We remember that delta G0 prime is not the determinant of a direction of a reaction, correct? The determinant of the direction of a reaction is delta G. Let's think about the delta G. There's your energy barrier. Delta G is equal to 23.9 plus RT times natural log of that whole product reactant business. Okay? How do we make delta G be negative, folks? We've got to make the log term more negative than 23.9. How do we do that? Well, one way is to increase the amount of reactant. The other way we do it is to decrease the amount of product. This is what I call pushing and pulling a reaction. Pushing a reaction means increasing the amount of reactant. Pulling a reaction decreases the amount of product. Now, I'm introducing this topic to you. I will show you this topic next time, but in the meantime, I think we're ready for a song. This is a fun song. It's one of my more popular ones. It's called The Sound of Glucose. Please join me. High sugars are always in ketosis. If there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Forget my own song. Onto a glucose we add a P to it. ATP energy ought to renew it. Quick rearranging recreates F6P without requiring input energy. At a high rate, add a phosphate with PFK. F16BP is made up this way so we can run and play. Aldolase breaks it and then it releases DHAP and a few G3 pieces. These both turn into 1, 2, 3 G, adding electrons onto NAD. Phosphate plus ADP makes ATP, while giving cells what they need, energy. Making triphosphates a situation of substrate level phosphorylation. PG to BPG, lose a water. PEP gets a high energy state just to make pyruvate. So all the glucose gets broken and bent. If there's no oxygen, cells must ferment. Pyruvate, lactate, our cells hit the wall. Some lucky yeast get to make ethanol. This is the end of your glucose's song, unless you goof up and get it all wrong. Fake it, don't make it to yield ATP. You'll save yourselves from futility. Okay, guys, see you on Wednesday.